So in terms of ordering molecular testing in patients with non-small cell lung cancer, I think there's a couple of components to that question, looking at both early versus late stage, as well as squamous versus non-squamous non-small cell lung cancer. So in general, I order molecular testing on the majority of my patients with non-small cell lung cancer. I order it on all my patients with advanced non-squamous non-small cell lung cancer because we know that there are great therapies for both FDA-approved drugs matching them to a particular mutation, such as EGFR mutation, ALK rearrangement, RUS1 rearrangements. We also have a host of new potential targets that are actionable either using off-label drugs or on a clinical trial in terms of expanded panels. For patients with squamous non-small cell lung cancer, if you look at the guidelines, they talk about ordering molecular testing if patients have a scant biopsy specimen or if they have a never or light smoking history. So I typically follow those guidelines and order the molecular testing on those patients as well, because I found in certain cases, it's uncommon, but it does happen in squamous lung cancer that's obtained on a scant specimen or a patient who has a very light smoking history. I found actionable molecular alterations, EGFR mutations, ALK rearrangements, and that's really impacted and changed the patient's course of care. And for patients with squamous lung cancer in general, I also order molecular testing, although EGFR and ALK EGFR mutations and ALK rearrangements in patients with squamous lung cancer is very uncommon. We do at UC Davis have the Lung Master Protocol, which is a Southwest Oncology Group trial that allocates patients to different treatment arms based upon their molecular testing results to look at other targets beyond EGFR and ALK because they're so uncommon in patients with squamous lung cancer. So on a trial, I do, even if the patient has a heavy smoking history with squamous lung cancer, on that trial I do order molecular testing because that could also impact their care as well. In terms of early versus late stage non-small cell lung cancer, and late stage I generally do it on everybody with non-squamous and non-small cell lung cancer, and patients with squamous that have those characteristics of never light smoking or a scant biopsy specimen to look for EGFR, ALK, and RUS1 in particular, but also that expanded panel as well. I'm doing it more in earlier stage patients, but that's basically in association with the Alchemist trial that we have open at UC Davis. So generally it's not standard. You don't have to do it on early stage patients, but because we have the Alchemist trial, which is an intergroup trial that randomizes patients with an EGFR mutation to receive erlotinib or placebo for 2 years, or with an ALK rearrangement, crizotinib or placebo for 2 years. Those are two of the arms in the study, and soon to be added an immunotherapy arm as well. I generally obtain of those patients because if I have something to offer them if it's positive. But I generally don't treat with EGFR TKIs, for example, in EGFR mutant lung cancer in early stage patients off of a clinical trial. So in terms of my standard molecular testing panel for patients with advanced non-small cell lung cancer, I generally send a next generation sequencing panel that has about, there's several different vendors, there's several hundred genes on these panels, but the main ones you're looking for are EGFR mutations, ALK rearrangements, and RUS1 rearrangements. So those are the big three. So no matter what molecular testing you do, if you do targeted testing, just having EGFR mutation, ALK rearrangement by FISH, RUS1 rearrangement by FISH, that is sufficient, but the NCCN guidelines note that there are potentially other actionable molecular alterations such as MET exon 14 skipping events, HER2 insertions, and others that could be either open up patients to get clinical trials and have those available to them, or that you potentially at some point in treatment you could use an off-label drug that may have targeted activity, such as for BRAF mutant lung cancer or MET exon 14 skipping events. So I generally start off with an NGS panel because I feel you all get all the answers at once, but sending off an EGFR mutation test as well as ALK and RUS1 FISH for the rearrangements is also sufficient. But then keep in mind, if those are negative, particularly if a patient has a never or light smoking history, to follow up with an NGS panel, after they've progressed on a platinum-based chemotherapy, for example, because you may pick up some other actionable molecular alterations that you can match to a trial or some off-label therapies. So in terms of the status of plasma cell-free DNA, I think that's an exciting new option in terms of molecular diagnostics that are available to our patients. So there are multiple different companies that offer these tests, as well as several academic institutions. And basically, it's looking at cell-free DNA in the blood that's shed by the tumor to see if we could pick up these molecular alterations. So there's been a lot of research, and there's still a tremendous amount of work that remains to be done, but it could be a useful adjunct to tissue testing. So I have used it particularly in patients with EGFR mutant lung cancer to look at the emergence of T790M mutations, which is the most common resistant mechanism to erlotinib, gefitinib, afatinib, your standard frontline EGFR tyrosine kinase inhibitors. You know, I'm generally using plasma cell-free DNA more and more, especially for detecting emergence of EGFR T790M, which is the most common resistance mechanism to your standard first-line EGFR TKIs, such as afatinib, gefitinib, and erlotinib. Although I don't think it necessarily substitutes for tumor tissue. It's a way of enhancing your detection for EGFR T790M and potentially accelerating that process, since you don't need to obtain a tissue biopsy, which can take some time as an invasive procedure in terms of getting that result back.